Hi, folks. Well, thank you, Sarah, for inviting us. It's very exciting to be here. So my name is Ricardo Albanus. I'm a postdoc at the Harvard lab at WashU in St. Louis. And I have here Gina Finan from Columbia University in New York City. And we're very excited to present our work here at the UKDRI Neurogenomics Seminar Series. So today we're going to talk about the work we're doing across the pond from you to characterize cellular crosstalk patterns in the brain to understand molecular mechanisms of Alzheimer's disease. So we'll define as crosstalk in this presentation any interaction between two cells that depend on a ligand in one cell and a receptor in another. And crosstalk is involved in normal brain physiology in processes such as synaptic pruning, as Beth Stevens' group showed over 10 years ago. But when we look in the more recent literature, we're seeing increasing evidence that crosstalk plays an important role in neurodegeneration, particularly involving microglia. And this includes the recent work from Francisco Quintana's group using the rabbit-seq tool. So I'm going to talk about this work just a bit because I think it's a good setup for what we're doing. So they use this barcode system to quantify crosstalk interactions involving astrocytes and then determine drivers of immune activation in a mouse model of multiple sclerosis. And they identify two crosstalk signaling pathways involving astrocytes and microglia as regulators of this neuroinflammation. And these are the plexin-semaphorin crosstalk and the afferent signaling pathways. And the reason I'm highlighting this study is that they were able to attenuate clinical presentations of this mouse model by using a small molecule targeting this astrocyte microglia crosstalk through the afferent signaling. So this shows us that looking at these crosstalk interactions can help uncover new therapeutical interventions for neurodegenerative diseases. But the limitation of most of the existing studies, including the Quintana group, is that they're focused on very specific questions, right? They either are looking at very specific pathways, or in the case of the Quintana group, they're focusing on a single cell type. And these are incredibly important studies, but there's, in our opinion, a need for a systems level understanding of the role of crosstalk in neurodegenerative diseases, particularly AD. So we wanted to zoom out and perform a systematic and unbiased characterization of the crosstalk patterns across all the cell types in the brain. And to this end, I'll be primarily focusing on our group's single-nucleus RNA-seq data, which has been recently described in a preprint. So this data set is comprised from parietal cortex tissue sample from 67 donors from the 90DRC and Diane cohorts. And it encompasses several neuropathological states across a wide spectrum of genetic backgrounds. And here's a bird's eye view of the data. So we assay almost 300,000 nuclei and then assign them to 7 major cell types. And within those cell types, many more transcriptional states. And for more details on the data set, you can refer to the preprint. And for the purpose of this talk, I'll be mostly focusing on the findings at the level of cell types. So our first step was to use computational tools to unbiasedly estimate crosstalk interactions across each donor category. And by that, I mean the different neuropathological states and genetic backgrounds. And in order to estimate the crosstalk patterns in the first part of the study, we used the tool CellPhoneDB from Sarah Teichmann's lab. So this tool uses a curated database of ligand receptor pairs and then predict crosstalk interactions based on the expression of these ligand receptor genes in pairs of cells, and then score them based on the gene expression specificity. And to give an overview of the type of data we obtained from CellPhoneDB, we perform over almost a million tests over thousands of cell state pairs to obtain tens of thousands of significant hits. And on the left is a high-level view showing that we detect crosstalk interactions across all major cell types, with some more prevalent than others, as you can see for the endothelial cells here. And this is just for the control group. So we need to do this for all the donors. So we performed this analysis in each donor category in a data set and then statistically tested for changes in crosstalk patterns. And as you can imagine, this is a ton of data to parse. So we'll start with kind of like the high level analysis. So I'll start first by showing the global changes in frequency in these crosstalk interactions in the sporadic ADs versus the controls. So on the y-axis, you have the main cell types, and they're colored based on how they change in crosstalk compared to the controls. And you can see that we observe more interactions involving microglias, either the emitter or the receptor cell, and fewer interactions involving neurons. And we do that for all the conditions. And you can see that there's some specific changes, but generally we can appreciate that the strongest changes of crosstalk occur in microglia. So next we looked at what type of biological processes these changes in crosstalk perturb. And again, we have the different donor categories on the y-axis, and on the x-axis we have different biological processes. And you can see that there are some shared processes like neuronal death and immune activation, which match our expectations for Alzheimer's. But some processes seem specific to each donor category, such as the afferent signaling in the autosomal dominant AD, and that's the pathway identified by the Quintana group. So we next try to narrow down our search space by looking, because this is still a ton of data. So we wanted to look at genes that were implicated in Alzheimer's disease based on GWAS and functional studies. So using a list of around 800 AD genes, we looked at interactions that have one of those AD genes as either the ligand or the receptor. So just to give a couple of examples, we have CMA6D and Plexin A1, which require TREM2, have APP and CD74, and a couple others. So the matrix on the left show how many of these crosstalk interactions involving one of those AD genes were significant in a pair of cell state. And I'll start by showing data from a single microglia transcriptional state. Here is the first column. And you can see that the highest frequency of these interactions is with excitatory neurons, but we see them across other cell types as well. And here is the full interaction matrix, and you can hopefully appreciate that immediately we see a high frequency of AD interactions involving microglia, right? This first big column here on the left of the plot. And if we look further, we see that a lot of these interactions involve neurons. And we also see a high frequency of those AD interactions in endothelial cells, which suggests that they contribute to AD. So next, we're looking at the statistical enrichment for those AD-related interactions. And again, we have the different cell types on the y-axis, the donor categories on the x-axis, and the color corresponds to the enrichment of a given cell type is more than expected from these AD crosstalk interactions than you would expect just on the total number of interactions. And you can see that they are by and far largely enriched in microglia. And at least for this data set, we cannot really say that endothelial cells are specifically enriched for these AD interactions. If you remember, they had a ton of interactions just baseline. But keep in mind that this is a rare cell type in our data, so we're likely underpowered to look at endothelial cells here. And we can take this one step further and test which cell type is statistically enriched to interact with microglia for these Alzheimer's disease crosstalk interactions. And very interestingly, we see that the neurons are the most enriched cell type to interact with microglia. And we did the same analysis in other brain single-cell RNA-seq data set, and we see very similar results. So here I'm showing the ROS-map data from the Colonna lab, which is a different brain region, a different cohort. And we see again that strong microglia enrichment for these AD crosstalk interactions, and the data suggests that neurons are the likely partners. And we see similar results in Nancy-Eaps and Vivek Swaroop's single-nucleus RNA-seq data as well. So to further confirm that the enrichment patterns we observed from microglia were specific to AD and not a result of some technical bias from the methodology, we decided to look at the genes associated with other neuropsychiatric traits and determine what were the enrichment patterns for these traits in our data set. So this is the same plot that I just showed in my previous slides. And to look at these other neuropsychiatric traits, we only look at the control group from our data set. And now we'll query using the controls different gene lists informed by several neuropsychiatric GWAS. And when we look at other traits, we see very different crosstalk enrichment patterns. So this is a busy slide, so let me walk you through it. We have on the x-axis multiple gene lists informed by GWAS studies, and they're clustered by similarity. So you see most of the Alzheimer's disease studies are close to each other, which reinforces the robustness of our analysis. And in addition, we see that other traits, such as Parkinson's disease in the right, have very distinct enrichment patterns. I won't have time to explore these results in depth, but it also shows that in addition to not being a method bias, we can extend this crosstalk framework to look at other diseases and do what we're doing here for Alzheimer's disease. So based on these results, we wanted to investigate what's the likely downstream mechanisms of the neuron-microglia crosstalk and prioritize biologically relevant interactions. So the limitation with CellPhoneDB, which was the tool that we're initially using to estimate these crosstalk interactions, is that it cannot go beyond indicating these highly specific interactions. It cannot tell you what's connected to these crosstalk interactions, cannot really give you an idea of effect size. So instead, we use this other tool called Cytokine that reconstructs the signaling network downstream of the crosstalk interactions using information theory. So it applies information theory to the gene expression data. And this is a completely orthogonal way to identify crosstalk interactions. So for these cytotoxic networks, we'll color code the genes based on which cell type they're expressed in the data. And the red border indicates whether it's one of those Alzheimer's disease genes from our list. So on the left, we have a subset of the ligand receptor pairs we identified previously with CellPhoneDB. And here's a reconstructed crosstalk network using cytotox. So you can hopefully appreciate that this gives us a very powerful way to query the biology of these crosstalk interactions and identify what's downstream of them. And what's important is that we see many of those red Alzheimer's disease-related genes in the microglia side of the network, which indicates that the crosstalk signaling between microglia and neurons is predicted to affect disease-relevant genes. And we test this statistically, and we see a very clear enrichment of these AD genes in the microglia side of the network, no matter which subset of the data we're using to reconstruct the networks. But on the other hand, we don't see enrichment for these AD genes in neurons. And what's important is that the crosstalk interactions that I'm showing here in the center, they're actually prioritized by Cytokine based on how they connect to central genes on the networks from each cell type. So initially there were 13 crosstalk interactions involving AD genes that were significant by CellPhoneDB, but we end up with only 2 being prioritized by Cytokine based on this network topology analysis. So we want to focus now on this crosstalk involving TREM2 and ThyroBP, which is the gene that encodes DAP12. And this corresponds to the plexin A1 semaphorin 6D crosstalk. And what's really interesting about this crosstalk is that it has been shown to induce macrophage activation in a TREM2-dependent manner. And in fact, it was identified using co-immunoprecipitation that TREM2, DAP12, and plexin A1.
It's okay. Yeah, good you are here. So I think he...
Sorry.
Have a question?
No, I think someone is accidentally unmuted, maybe.
All right, I'll carry on.
Yes.
So the cool thing, keep in mind here, is that using co-immunoprecipitation, they found that TREM2, DAP12, and plexin A1 likely form a complex, which I'm showing on the right. So we think we're picking this. And more broadly, plexin semaphorin signaling has been implicated in immune modulation. So there's a TREM2 dependent role in macrophage activation that I mentioned in the previous slide. So this is showing that this is wrong. This should be SEMA6D, not 6A. This is a typo. We're showing that once you treat macrophages with SEMA6D, you will increase cytokine secretion, right, in a TREM2 dependent manner. And the same plexin So plexin A1 with a different semaphorin, SIMA3A, was linked to microglia activation. So this is getting closer to the neuroinflammation aspect. And more recently, the Quintana group paper that I mentioned in the intro-identified plexin semaphorin crosstalk as a modulator of neuroinflammation. So this together makes this family a really attractive target to study further in the context of neurodegeneration. Now, back to the crosstalk networks, we wanted to look at what's connected to this TREM2 plexin semaphorin crosstalk in microglia. And to do that, we use network algorithms to extract subnetworks and make it easier to explore the data. So we're not looking at this big hairball here. And if we pick this apart, here's the TREM2 subnetwork. And you can see it's predicted to propagate signals into several Alzheimer's disease-related genes, including ApoE and genes from the HLA family. And if we do a functional enrichment, we can see that it's involved in immune recognition and activation. So this means that we're capturing functional aspects of microglia biology with this unsupervised approach. And it's independently recapitulating the link between TREM2 and microglia activation. And on the right, we applied the same approach to Nancy Ip's group single-nucleus RNA-seq data, and we're able to find a very similar subnetwork in microglia. And we see this subnetwork or parts of it in the Swarup and the Colonna data set. So this is indicating that this is a highly reproducible aspect of microglia biology. So now we wanted to leverage our large cohort and determine the association of this TREM2 subnetwork with neuropathological burden in AD. So our data set is fairly well represented across Braak stages. So this way we can correlate the gene expression of these subnetworks with Braak stage. So on this plot on the left, the y-axis is the association of the subnetwork gene expression with Braak. So positive values mean that the expression of this network is positively associated with Braak, and therefore it's predicted to increase AD risk. And negative values means that it's predicted to increase Alzheimer's disease resilience. And I'll start by showing a subnetwork that has the presenilin-1 gene, which is a well-known player in AD. And you can see, as expected, it's positively correlated with Braak stage. We look at another known player, APP. Again, this is not just the APP gene, but rather the network that encompasses APP. And you can see that this network is positively correlated with Braak stage. And here are all the microglia subnetworks. And you can see that our TREM2 subnetwork is among the most negatively correlated with Braak stage, which suggests that it has a protective role in AD. And if we flip sides and do the same thing in neurons and look at the SIMA6D subnetwork, you can see that it agrees in the direction of effects, suggesting that this crosstalk as a whole has a protective mechanism in AD. And in my previous slide, I cheated a little bit because I showed all the co-expression subnetworks in microglia. It was not just the cytotalk ones. So here is just the crosstalk subnetworks, the one that were identified with cytotalk. And we can see a different pattern more clearly now. By and large, the genes involved in the neuron-microglia crosstalk are predicted to be protective in AD. So this suggests that crosstalk dysregulation is an important component of Alzheimer's disease biology. And now I'm going to show some unpublished data, some work that this is still work in progress. So we are looking at spatial transcriptomics data from human brains to further understand how these crosstalk interactions are disrupted in AD. So this is a collaboration with Dr. Hongju Fu lab at Ohio State School of Medicine. And they generated spatial transcriptomic data from 3 cases and 3 control brains, and they have the corresponding immunostaining data for beta amyloid. And this way we can correlate gene expression with disease burden. So in this plot on the bottom right, each circle corresponds to 1 spot in the Visium platform. So these are our units of gene expression. And the color corresponds to the proximity of each spot relative to the A-beta plaques, So level 1 is 1 spot away, right? So blue here is the plaques. You can see them here on the left. And level 1 is 1 spot away, level 2 is 2 spots away, and so on. And this data was all manually annotated by Shua Shane. So I cannot overstate the amount of work that went into this analysis. And if you're familiar with this data, you know that this is not really single cell resolution. So each spot can have multiple cells, and this will lead to some challenges regarding differential gene expression downstream, which we're still trying to solve. So we did several analyses, but in the interest of time, I'll only show a couple of results. So the first thing we see is that there are more microglia positive spots in the cases versus control, and this is roughly expected based on previous literature. And when we look at the distance to plaques, however, we don't see significant difference in the number of microglia when comparing the plaque positive versus negative spots in the Alzheimer's disease in the cases, right? So this is suggesting it's roughly uniform. And next, we looked at the spots that are positive for microglia and checked how that TREM2 subnetwork behaved in proximity to the plaques. So each point in this plot corresponds to the gene in the TREM2 network. On the y-axis is the log2 fold change relative to the control. So positive values means that this is higher in whatever group we have on the x-axis. So in this case, this is the plaque overlapping spots, and you can see that they're higher. And as you can see, this network is upregulated in Alzheimer's disease, but interestingly, it's becoming weaker the closer you are to a plaque, right? So this aligns. So when you look at the spots that are further away from the plaques, that's where we see the highest expression of this TREM2 network. So this aligns with our previous results in the single-nucleus RNA-seq data and suggests that this network is getting exhausted, if you will, with a higher neuropathological burden. And as I mentioned previously, the differential expression analysis are challenging with this data, so we cannot really test, at least not now, whether the SIMA6D activates a TREM2 network with the current approaches. And I'm happy to chat more offline about the technical limitations here. But the next best thing we can do is look at microglia positive spots and determine the distribution of microglia spots where we also detect SIMA6D. So this can give us clues about whether microglia are attracted to cells expressing SIMA6D and how this changes during disease progression. And we tried looking at this data from multiple perspectives, and the clearest patterns we saw was when looking across cortical layers. So here in this plot on the y-axis, we have a fraction of SIMA6D spots when comparing all the microglia spots, right? So these are microglia with SIMA6D. And you can see differences in cases and control. First of all, just ignoring the color labels, we can see that most of the SIMA6D positive spots tend to be in the middle cortical layers. But when we look at a difference between cases controls, you can see difference. And particularly, we see a decrease in microglia near SIMA6D positive spots in these middle layers, but we see a massive increase in microglia getting closer to SIMA6D expressing cells in the white matter. And we're still trying to figure out why that happens, and we're using more advanced computational tools to query this. But the take home to this slide is that to some degree, and SIMA6D homeostasis, and I'll use that with quotes, is disrupted in AD. So all of this gives us some interesting circumstantial evidence regarding the role of the TREM2 plex in somatoform crosstalk in AD. But at the end of the day, we need more information. And to gain further insight on the molecular biology associated with this crosstalk signaling, Gina will talk about the work she has been doing on iPSC-derived microglia. So she was working separately on this question from a completely different starting point, and we're very lucky to learn about each other's work doing AIC last year, and this motivated us to collaborate on this project. So Gina, please take away.
Thanks. Okay, so our lab is interested in investigating microglia TREM2 signaling in the context of Alzheimer's disease. And the contribution of microglia in regulating Alzheimer's disease pathology is gaining significant traction with the identification of TREM2 as a significant genetic risk factor for Alzheimer's disease. So in this slide, I'm showing that deleterious stimuli known as disease-associated molecular patterns, such as A-beta, can induce a homeostatic microglia to become activated and trigger certain functions, such as phagocytosis and cytokine release. So such acute activation, can rid the surroundings of the assault, and the microglia can resolve to its former homeostatic state. However, upon a sustained assault, sustained A-beta exposure, the microglia can become chronically activated, resulting in chronic inflammation, impaired phagocytosis, and leading to neurotoxicity and synaptic dysfunction. Okay, next. Okay, so it's important in our studies to use a physiologically relevant cell model. So as shown in figure A, we used microglia differentiation protocol developed in the Blurt-Jones lab at the University of California, Irvine. And so the iPS cells undergo hematopoiesis using a commercially sourced cytokine and growth factor optimized media. And the resultant CD43 positive hematopoietic gender cells are then differentiated with the addition of MSCF, IL-34, TGF-beta, and insulin, and mature with further addition of CD200 and CXCL1. So there below you see bright field images showing the progression from day 0 to day 34, in which the iPSC colonies grow, and at day 7 begin to release the hematopoietic progenitor cells into the media. And finally, the last bright field image, you see the mature microglia. So in figure B, we're showing immunocytochemistry and expression of common microglia markers, TREM2, IBA1, and TMEM119, counterstained with DAPI. So next. So here in figures A and B, we're showing A-beta and synaptosome phagocytosis, and that these microglia are able to phagocytose proto-labeled A-beta and synaptosomes. So proto is a red dye conjugate that only fluoresces in the low pH environment of the phagosome. So figure A is co-stained with a cell surface marker in green, and you see the phagocytosed A-beta particles. And in figure B, it's showing a masked synaptosome uptake with a fluorescent and phase contrast imaging overlay. Figure C demonstrates that our microglia are responsive to LPS, and LPS is a commonly used pro-inflammatory stimulus for microglia. And this is showing cytokine release of IL-1 beta, IL-6, IL-8, and TNF-alpha as measured by a mesoscale immunoassay of pro-inflammatory cytokines. So you see with addition of LPS, a marked increase of the cytokine release. And this is mitigated by addition of bicytine, which is a compound that inhibits inflammatory response. And then there at the bottom, I'm showing some bright field images of plus and minus LPS addition, just showing some of the morphological changes in the microglia. Next. So to facilitate our studies into TREM2, we generated a CRISPR-modified iPSC-derived TREM2 knockout microglia. And we used a vector from the David Feltzer lab at UPenn to facilitate our targeted gRNA delivery against TREM2, leading to a frame shift deletion. And then we are showing there on the right, Western blot analysis demonstrating a knockdown of our TREM2 expression. And in figure B, we measure phagocytosis by our microglia, the wild type versus knockout, which is the blue versus black. And we see about a 55% decrease in TREM2 knockout cells phagocytic activity. And that blue, the light blue and light, or the gray, the light blue and gray points are cells treated with cytokine D, which is an inhibitor of phagocytosis, and see that cytokine D does indeed inhibit phagocytosis of both wild type and knockout. Next. So, whereas Ricardo's discovery, so like Ricardo mentioned, we met at the Alzheimer's Association conference, and so his discovery of SEMA6D and TREM2 association using genomics, transcriptomics approach was fascinating, and our lab used a molecular biology approach to examine a hypothesis basically that began with a literature search into TREM2 signaling, in which we found that TREM2 forms a receptor complex with plexin A1 and DAP12, in which SEMA6D binds to plexin A1 to induce immune responses in dendritic cells and macrophages. So naturally, we wanted to investigate this system in human microglia. So we used Turbo ID to verify the association of the plexin A, TREM2, DAP12 components as expressed in 293 cells, knowing that our microglia express all of these proteins. And what this does, if you see in figure A, is that this approach tags a protein of interest with Turbo ID, which is a biotin ligase. And upon cell treatment with biotin and ATP, Turbo ID enables biotin labeling of proteins within 10 nanometers. So the proximal proteins can then be pulled down with streptavidin beads, which we see in figure 5. The left side of those two panels, you see the input expression of our proteins that we're looking at, that are vectors 1 through 6, that are listed on the right. So vectors 1 and 2 are negative controls, GFP, these are all flag tagged, and empty vector, and vectors 3 and 4, which express plexin A1 and plexin A4, which are two different family members that are known receptors of SEMA6D. Vector 5, an unrelated protein, and vector 6, which expresses DAP12 as our positive control. So on the left, you see the input of all of those with plexin A1, plexin A4, GFP and DAP12, and NRP1. And on the left-hand side of the right panel, the input of TREM2. And then we see the streptavidin elution on the left panel on the right-hand side. Indeed, our streptavidin pulls down plexin A1 and plexin A4 and DAP12. And then we see, of course, the pull down of TREM2, mature TREM2. Next slide. So we wanted to test the effects of SEMA6D on our microglia by measuring phagocytosis with fluorescent imaging, and found that SEMA6D increases phagocytosis in wild type cells by about 40%, and these are our blue points. Treatment of TREM2 knockout cells, the black points with SEMA6D, however, elicited no added responses with any of the varying concentrations, 10.5 or 2.5 micrograms per ml. And since no SEMA6D concentrations further increase phagocytosis in the TREM2 knockout cells, this is suggesting that SEMA6D is signaling through TREM2, and phagocytosis of synaptosomes is dependent on TREM2 signaling. Next. So we also wanted to see the effects of SEMA6D on pro-inflammatory cytokine release in our microglia. So we see that SEMA6D, in fact, increases TNFα and IL-6 in wild-type microglia, but elicits no response in our TREM2 knockout cells, as measured by mesoscale multiplexed immunoassay. So dysregulation of inflammatory cytokine signaling is implicated in the pathogenesis of Alzheimer's disease, which activated microglia might have good or bad effects, depending on acute versus chronic activation, as we saw in the first slide. But it has been found previously that TNF-alpha has been inversely correlated with neuronal degeneration and apoptosis, and that IL-6 in vivo studies suggest that IL-6 can activate microglia and enhance A-beta phagocytosis, all of which suggesting potential therapeutic effects of SEMA6D on AD pathology. Next. So in summary, iPS cells derived TREM2 knockout microglia inhibit phagocytosis of human synaptosomes. TREM2 associates with plexin A's and A1 and A4 and DAP12 via proximity labeling. SEMA60 increases phagocytosis in microglia in a TREM2-dependent manner. And SEMA60 increases TNFα and IL-6 cytokine release in a TREM2-dependent manner. Ricardo?
Right. So in addition to what Gina showed here, they also generated RNA-seq, bulk RNA-seq data for this treated and untreated microglia in the wild type and TREM2 background. So to end this presentation, I'll show the changes in gene expression associated with SEMA60 from this iPSC-derived microglia. So our first plots show the fold changes for all the tested genes in the bulk data. And you can appreciate that the SEMA60 treatment induces broad transcriptional changes in the wild type background, but not on the TREM2 microglia. And on the right, I'm showing the absolute effect size distribution of these log fold changes. And you can see that the treated wild type microglia have this very strong effects, and these are ablated when you have a TREM2 So we can go back to our TREM2 network and look how it behaves on this RNA-seq data. So each row is one of the comparisons, right? So SEMA60 treated versus control in the wild type in the KO, and then just comparing the TREM2 KO versus wild type non-treated. And each point in this data is one of the genes for the TREM2 network. And you can see that SEMA60 treatment on the wild type activates this TREM2 network, and these effects are mitigated when you're on the TREM2 background. And on the right, I have the heat map version of this plot where I'm just showing the median effect size for each of these comparisons. And I'm doing this so we can compare it to other gene signatures. And what's important, we have multiple gene signatures here on the x-axis, but in the interest of time, I'll just focus on one, is the second signature. These are genes that are upregulated in phagocyting microglia. They're also upregulated under SEMA60 treatment. So this aligns with the increase in phagocytosis that Gina experiments showed previously. So to wrap up everything here, we are showing that plexin A1 semaphorin 6D interactions modulate microglia activation and are likely protective in AD. And this provides a broader support for the role of plexin and semaphorins in neurodegeneration. We identify neuron-microglia crosstalk as an important component of Alzheimer's disease, which can propagate signal in microglia and affect other downstream AD-related genes. And more broadly, the cellular crosstalk network approach can help understand the biology of Alzheimer's diseases and other neuropsychiatric traits and identify new drug targets. So this is using functional genomics to contextualize, give an additional layer of context to GWAS-nominated Alzheimer's disease genes into these biological pathways. And even more broadly, this what we're doing here is a robust analytical framework that can be used to generate novel biological hypotheses. As I showed the previous results with the other neuropsychiatric traits, we can essentially apply the same strategy to look at other diseases. And if you want more details about this work, you can check our preprint on bioRxiv. And with that, I want to thank our large group of collaborators, and most importantly, the donors and their family for making this work possible. And here are Gina's acknowledgements. So thank you very much.